Hello dear learners. In the previous video, we discussed about the protein chemistry, the content of that particular unit or the subtitles that are included under the heading of protein chemistry. Now we are going to discuss about the second unit, that is biochemistry and molecular biology techniques. This course is also for the 15 credits, and the basic idea behind this unit is you have to study about molecular techniques which are very much important for the molecular biology and biochemistry. Like MBBT, molecular biology and biochemistry technique is a separate topic, and it is very vast topic in the field of research. So first of all, we will discuss about the headings. फर्स्ट इज योर क्रोमेटोग्राफी. क्रोमेटोग्राफी में हमें पढ़ना है प्रिंसिपल्स एंड एप्लीकेशन ऑफ जेल फिल्ट्रेशन, आयन एक्सचेंज एंड एफिनिटी क्रोमेटोग्राफी. Chromatography, it is a technique of separation of molecules on the basis of charge, on the basis of size, on the basis of various properties. So for this purpose, chromatography, we have to study about principles. What is the principle behind the chromatography technique? What are the applications of chromatography technique? And we have to discuss about different types of chromatography like gel filtration, ion exchange chromatography, affinity chromatography, and we have to study about the principle, methodology, applications of these three different chromatography technique. Next one is your electrophoresis. Already in the TY-BSc microbiology, you gone through this technique, electrophoresis. It is used for the separation of molecule on the basis of charge and size. Like you studied about the isolation of DNA or the DNA gel electrophoresis. You also study about the protein separation by Western blotting technique, or we also separated the DNA by using Southern blotting technique. So for this purpose, in the electrophoresis, we have to study about gel agarose electrophoresis, native page electrophoresis, SDS page electrophoresis. SDS page is nothing but the sodium dodecyl sulfate page is for polyacrylamide gel electrophoresis. So electrophoresis is nothing but the technique of separation of molecules on the basis of charge and size. In that, we have to study about the types of electrophoresis, agarose, native page and SDS page. Next one is your polymerase chain reaction, that is PCR. In this, we have to study about the principle of polymerase chain reaction, variations of PCR like the types of PCR, hot start, nested, reverse transcription, that is RT-PCR, and then real-time PCR, qPCR, and its application. So polymerase chain reaction, what is the importance of polymerase chain reaction? We all are knowing because of this pandemic, COVID-19. All of the labs that are approved by the ICMR, they are performing the RT-PCR technique. So why they are going to perform the RT-PCR? It is very simple. The principle of polymerase chain reaction is the amplification of your interested molecule like DNA, RNA. Okay, so this is the polymerase chain reaction. And what is the principle of polymerase chain reaction? What are the types of PCR? We have to study the principal, applications, and the main methodology, and what are the enzymes that are used to carry out the process of polymerase chain reaction, and what are the different applications in different areas we have to study in this topic. Next one is your sequencing methods. Sequencing methods, like when there is evolution, we are knowing about the evolution. This is done by the sequencing method, like 16S RNA sequencing method or RNA sequencing methods. What are the applications? DNA sequencing method, classical and next generation sequencing method that includes pyrosequencing, ion torrent sequencing and nanopore sequencing. So what are the applications, principle, methodology to carry out these processes we have to study in the sequencing method topic. So if you have any doubts related to this topic, you can ask, and also go through this basic concept. What are the chromatography, what is the principle? When we are going to start about the lecture, it will help you to recollect the information. Thank you.